This is chapter 10. This is the start of the nervous system. So let's first talk about the tissue, the cell types. But the nervous system, as we already know, it will detect changes, help make decisions, stimulates muscles and glands to respond, and helps maintain homeostasis in the body. There are two types of cells. We have neurons and neuroglia. We've looked at these when we talked about nervous tissue, but we'll spend more time on it today. The neurons are your main cell body that reacts to changes and sends nerve impulses for communication. Neuroglia are supporting surrounding structure which is nerve tissue. It provides nutrients, and it also helps protect the blood-brain barrier. We divide our nervous system into the central nervous system, or the CNS, and the peripheral nervous system, or the PNS. The central nervous system has the brain and the spinal cord, and the PNS has the cranial nerves and spinal nerves. Here's some images of them. The division of the nervous system with our central nervous system can be the brain and the spinal cord. And then peripherally, we have the sensory division and a motor division. Sensory division sends information to the brain and spinal cord. Motor division is bringing a response from the brain and the spinal cord back out to muscles or glands. And you can divide that further into somatic nervous system and autonomic nervous system. Somatic is going to be going to skeletal muscle. Autonomic is going to be going to smooth muscle, cardiac muscle, and glands. Some of the sensory function of the nervous system. Nervous system receives information. It will gather that information and by detecting changes, and then it sends it to the central nervous system. Inside the CNS is where the integrative function will occur, which means it takes that sensory information, makes a thought or a decision about it, and then has a response or a decision to it. That response will be then the motor function that's acted upon. That signal will then be sent down a neuron and carried to an effector, which is always going to be a muscle or a gland. Again, we have our somatic nervous system, which is going to be our voluntary instruction to the skeletal muscles, and autonomic, or automatic, you could consider it, but it's called autonomic. That transmits involuntary instruction to our CNS, or from our CNS to smooth muscles, cardiac muscles, and glands. So the neurons are kind of like the cell structure we learned back in chapter 3, but they will vary in size and shape. They have some certain features. There's a cell body. Sometimes that's also referred to as a soma. It contains a nucleus, cytoplasm, organelles, and some neurofilaments, as well as this granular substance called chromatophilic substance, or Nissl bodies. That's a unique feature. Only in the neuron will you find that. Now, off of the cell body are projections called dendrites, and they are branched. It's a receptive surface, meaning it's gaining information. And the neuron could have many of these dendrites. An axon is a projection on another end of the cell body that transmits impulses and releases neurotransmitters or chemicals to another neuron or effector. And the neuron may have only one axon. So here's a picture of a typical neuron. We have our cell body, and see the green chunky stuff up there? That's our chromatophilic substance or Nissl body. We have our dendrites. That looks like hair hanging off of it. And here's our axon, that long tail off of there. And the axon starts off of the cell body called the axon hillock. A hillock is basically like a hill or a rise to a hill. And then it extends into the axon. Now this picture of an axon is one that we would say is myelinated. What that means is it has this fatty substance that is wrapped around the axon, and those that they're actually in cells. So these fatty little bundles are called Schwann cells. Inside the Schwann cell, it's made up of myelin, which is a fat-like substance, as well as a myelin sheath. In between those cells is a space that is bare, and that's called the node of Ranvier. That is a space intentional for conduction of the signal. We'll talk about that a little later. At the end of the axon is the terminal end, meaning that's where it stops, and it has a knob at the bottom called a synaptic knob. Here is another picture in microscopic version, as well as another one. So our Schwann cell is only going to be in the peripheral nervous system. So we can label it as peripheral nervous system neuroglia. And it encases axons in a sheath, or it wraps around it. These images are good because it shows you how it kind of wraps around, kind of like insulation. It's composed of a lipoprotein, so a fatty protein called myelin, and that coating is called the myelin sheath. And in between them again is that gap called the nodes of Ranvier. So not all axons are myelinated. If it's myelinated in the PNS, it will have a series of those Schwann cells lined up along the axon, and each having a wrapped coating of myelin insulating the axon. And this top picture shows us a myelinated axon. The bottom is unmyelinated. This is common also. There's unmyelinated axons. And they are encased by a Schwann cell cytoplasm, but no wrapping of myelin around the axon. There are different types of neurons depending on their shape. The multipolar neuron is this one, the most common one we see, usually drawn or picture of. This is 99% of your neurons. There's many processes or dendrites, and there's one axon. So most of the neurons in the CNS are going to be multipolar. Bipolar, you can see there's two processes, an axon and then one dendrite. So it kind of looks similar from one end to the other off of the cell body. This is going to be for more specific small functions. So your eyes, your ears, your nose, those are going to be bipolar. And then unipolar is one projection off, and then part of it splits to be a peripheral, and part splits to be central process. And the cell bodies are in what's called a ganglia, a grouping of cell bodies. This is always going to be a sensory nerve. Here is our movement or further description of how neurons function. So think about, for instance, you are stepping on a tack. So there's a receptor in the bottom of your foot that's going to feel that sharpness. That receptor sends information up a sensory neuron to the CNS. So this dotted line is our division between our PNS and CNS. So when sensory information is going up that neuron, we call it a sensory neuron or an afferent neuron. Transfers the sensory information to the CNS. In the CNS, this neuron, called an interneuron, will then transfer that information from one part of the CNS to another, and then a motor neuron is sent out through the CNS to the PNS to send a signal. The motor neuron is also called an efferent. Think E for exiting the CNS. It's efferent neuron transfers information from the CNS to the effectors. And a effector is always a muscle or a gland. Now we're going to talk about neuroglia. The general function of neuroglia is to provide structure and support. In an embryo, it guides the neurons into position, and it can also may stimulate specialization. It produces growth factors to nourish neurons and remove excess ions and neurotransmitters, and it aids in the formation of synapses. So we have different names of them. An astrocyte is the first one. This connects neurons to blood vessels. And so astrocytes function as part of your blood-brain barrier. Your blood-brain barrier is like this barrier between your blood and your brain, and it helps keep certain chemicals out of your brain for protection. Astrocytes also regulate ion concentrations such as potassium. I'm going to jump ahead a second and show you a picture. So here's an astrocyte. You can see its little projections onto the capillary here, and then to another neuron. Oligodendrocytes is a fun word to say. It is a cell in the CNS that does myelination. It provides structural support. So here is a picture of our oligodendrocyte making myelin to myelinate these axons in the CNS. So it's similar to a Schwann cell, but this is in the CNS, so it's an oligodendrocyte. Schwann cells are in the PNS, so there's a big difference there. Microglia are your little janitor in the body. They are a phagocytic cell, meaning they are going to phagocytize or gobble up, engulf anything that shouldn't be in there. So here, look, it looks like a broom almost, or a Roomba. This is a microglial cell. It's going to go around and clean up debris from injury or broken down cells. And epididyma or epididymal cells are cells that line the central canal of your spinal cord, as well as these spaces in the brain called ventricles. They cover also a capillary structure in the brain called the choroid plexus. And these cells are specialized in helping regulate the composition or the making of cerebrospinal fluid. They are ciliated, so we can see a picture here. Here's your epididymal cells, and they're going to help make cerebrospinal fluid. Those are all in our CNS. Now, in the PNS, again, are Schwann cells, which produce myelin sheath around some of the peripheral axons. And the purpose of these myelination is to speed up the signal. When a signal is conducted from the cell body down the axon hillock, it will travel the length of the axon. But with these Schwann cells in here, that signal will literally jump over one Schwann cell to the node of Ranvier and to the next one. This increases the speed greatly. When there's no myelin on here, it has to travel the length of it, and so that signal is a little slower. There's also in our PNS a structure called a satellite cell. It supports clusters of neuron cell bodies or ganglia. Now cells nerve cells can die just like our epithelial cells do, but they function quite differently in some other ways. For instance, mature neurons do not divide. So this is why trauma to your spinal cord can be permanent. If a cell body is injured, the neuron usually will die. So some neurons can have regeneration out in the PNS. If the axon is injured, it could regenerate depending on how far out it is. If it's close to the cell body, it can cause death, but if the axon is separated from the cell body and its myelin sheath, it will degenerate. The Schwann cell and the neural lemma, which is like the outer layer, will remain, and then remaining Schwann cells will provide guiding sheath for a growing axon. So a growing axon can establish a former connection, so it will almost like regenerate itself. Function will return, and if not, the function may be lost. So it's not a set deal. Sometimes it grows back and works, and sometimes it doesn't. If you have damage in the CNS axons that lack that neural lemma to act as a guiding sheath, so oligodendrocytes do not proliferate after injury, and regeneration is very unlikely in the central nervous system. That's why spinal cord injuries are very damaging. So here's showing that regeneration in the PNS. So you can see on the axons further down, it's not close to the cell body. It's been damaged, and those Schwann cells will degenerate, but the neural lemma is still there, and so it regenerates and forms new cells, and then it will kind of connect back with that for the previous axon. That is chapter 10. Have a great day.